Are you there? Hi, Rachel.
Good morning, Lou.
Good morning. I've got a Monique.
Yes, you've got me.
How do I make you a co-host? How do I make you a co-host?
Okay. What you need to do is at the bottom where it says participants.
They're still joining us, and I know it takes a moment for the video and audio to catch up, but we'll get started now. This is our first of what I hope will be 4 or 5 course content reviews for AP Biology. You are aware of the unique structure of the exam and the course, and we're really I hope you are all taking the time to look at the College Board materials, but we are privileged to have presenters who understand the exam, and even though no one can predict what this year is going to bring, we'll help you do really content-focused preparation so that you can score as well as possible. And our presenter for today is Mary Beth Cook from Harrison County High School, who is an AP Bio reader and one of my tutors in AP Biology. She is one of my go-to people when I have a biology question. From the Advanced Kentucky side, Tina Rose is the English director. Lou Akampora is me, I'm the science director, and Aaron Timmons will be the math director. Those of you maybe taking other science or math or English courses may know them. And Monique Rice is our director for computer science and mathematics and helping me from the technological end of things. Please, as we go through this, mute your microphones and stop your video. Use the chat box. We will be monitoring the chat box, and please be as participatory as possible. We would love to hear questions coming from you. This is why we're here. And for any further information, there's a website, KYAP 2020, where you can find the recordings of these sessions and other hints or information. And without further ado, I'll turn this over to Mary Beth Cook, who will be presenting our lecture on, I guess, the structure and function of lipids and proteins specifically. Okay, Mary Beth, I think it's over to you, so you can share your screen if you'd like.
Okay, does this look okay to everybody?
Are you sharing your screen? There you go.
Okay.
I think it's coming up.
Hopefully. Slowly. I'll go ahead and give a shout out. I saw some of you were from Wayne County, so that's where I grew up. I went to Wayne County High School as well, so glad you could join us. Okay, I'll see if I can go ahead and get started, maybe. I think it's still loading. Okay. Does that look okay on your end, Lou?
Looks good.
Okay. All right. All right. So our focus today is going to be on structure and functions of macromolecules, specifically lipids and proteins. And so why are we talking about this? They do play such a huge role in our cells functioning correctly. And so organisms have to exchange matter with their environment to grow, reproduce, and maintain organization. And so as we look at structure, we'll also be discussing the elements that they're composed of. And so for living things, carbon is going to be that main element. And if you see carbon, so if they're showing you a structure of a molecule on a question and you see carbon with hydrogen, they may call these organic molecules. So in your head, you want to think, okay, so they consist of carbon and hydrogen. The main macromolecules, organic molecules that are important to living things are carbohydrates, lipids, nucleic acids, and proteins. And so, like I said, we'll focus on just the lipids and the proteins. So what makes carbon such an important element? It has 4 valence electrons. And so that means it has 4 electrons in its outer shell. And so it can either bond to itself or other elements like phosphorus, nitrogen, hydrogen, oxygen, and sulfur. And those are going to be elements that you're going to see in these macromolecules as we continue. And those 4 electrons can form covalent bonds, so really strong bonds where they share those electrons with itself or these other elements. And so because 4 bonds can form, molecules can be really large and they can have lots of shapes. And so shapes are going to really be important to the function of those molecules. All right, so here it is in wording in case somebody is looking at this at a later date instead of being live with us today. I want to go ahead and jump into some pictures of how these small units, these small molecules can form these large macromolecules that we're going to discuss. So I liked this picture initially, and then when I was reviewing and going through, I realized there was a spelling error, so please forgive that. I still like the picture. So monomer is a small unit, so mono means 1 if you're seeing that prefix, and that's going to be our building block to make these really large macromolecules. And so in this case, so this is just very generic, but monomer 1, so 1 small oval green circle, we want it to join to monomer 2, and then that's going to create our larger polymer molecule that consists of both of the monomers. And so to do this, to be able to walk through how does this actually occur, monomer 1 is going to supply this hydroxide group, monomer 2 the hydrogen, and so 2 hydrogens plus oxygen makes water as a byproduct. So that's released, and now monomer 1 can bond to monomer 2 through a covalent bond to create this larger structure. The other word that I want to mention here is that you might see in a question stem is an anabolic reaction. And so I don't know about you, but I've always had a hard time of thinking, what does anabolic mean, or even remembering what it means. And so through my studies, I always tried to find, whatever my biology term word started with, if I could think of a word that started with the same thing, that tells me what it does. And so A for anabolic, think of adding. So you're adding these small units together to make a larger molecule. All right, the opposite of that is sometimes we start with a really large molecule and we want to break it down. So we want to use those smaller components. And so the reaction that's going to allow that to occur is a hydrolysis reaction. And to break that down, if you're seeing hydro as a prefix, usually you can think of water, and then lysis means to break. So now we want to break those covalent bonds to go from a large molecule to those small individual components. And so you're seeing that in this picture. So to break the bond that's occurring between 3 and 4, water is inserted. So monomer number 4 gets the OH, and monomer number 3 gets the hydrogen. Another way that they could mention this in a question stem is use the term catabolic reaction. And so C for catabolic, think of cannonball. And so cannonball breaks a wall, water is going to help break that bond and separate those monomers. So let's try the chat out and see how this works for us today. If I had a polymer that was 10 monomers long, how many water molecules would it take to separate all those 10 units? Does anybody know how many waters it would take to separate 10 monomers? All right, so we'll go ahead. So Kelly Morgan's agreeing with Lou. Okay. All right. So kind of think about that. If you got 10 units together, it's going to need 9 bonds to hold them together. And so therefore it would take 9 waters to break those 9 bonds. Again, my second question, some of you did go ahead and answer that. What bond is holding monomers together? So I do kind of want to stress that they are covalent. Good job. All right, so all of that was kind of generic. Now we're specifically going to look at lipids. And so lipids are not technically a polymer like I just showed you, where you have a monomer connected to a monomer connected to a monomer. They're just really large molecules on their own, and they can consist of a variety of components. And you're going to see that as we move forward. Usually the smaller components are a glycerol and a fatty acid. And if they're mentioning some lipid that you've never heard of before, something I would want to pop in your mind is lipids are hydrophobic. And so again, if you look at the parts of the word, if you don't remember what hydrophobic means, hydro is water. And then if you have a phobia, you have a fear or a dislike. So lipids dislike water. If you've ever mixed oil and water in baking, you know they do not interact well. And then another word that goes along with this is the elements, carbons, hydrogens, and oxygens that we find in fats or lipids. They share those electrons equally. And so we're not going to see charges on those molecules. And because of that, they're not going to interact well with water because water does have charges and it's polar. So if you remember water structure, oxygen has a partial negative, hydrogens have partial positives. And so since fats don't have charges on their structure and water does, they do not interact well together. So here's some examples. I'm going to show you a triglyceride just so you can see structure and function, phospholipid, and then really quick, I'm going to mention a cholesterol. So just to tie what we've discussed so far to the fats, here's our small unit. So we have our glycerol, and then 3 fatty acid chains. So lots of carbons and hydrogens. That's where the name comes from. So the tri is the 3 fatty acid chains. Glycer, that part of the word comes from your glycerol. And so again, to have all of this come together as one big unit, one big molecule, water has to be removed. So dehydration reaction occurs, and we have our typical fat molecule that you're familiar with. So if we're talking about structure and function, what does that big molecule do? It's really great at storing energy for us. And so if you think about it, there's lots of chemical bonds to be broken, and all of that would provide energy if we needed it. So insulation, they can also provide protection to our organs, and we store fat in our adipose cells. All right, the next fat that I want to discuss is a phospholipid. So some of the parts are still the same. So we still have a glycerol here, and then we only have 2 fatty acid chains. And so if you're looking at this, so phospholipids, I just want to make the connection that it is making up the majority of your cell membrane. And what's the function of the cell membrane? That's to regulate what gets to come and go from the cell. Excuse me, I'm going on 5 weeks of isolation. I haven't talked that much. So anyway, back to these fatty acid tails. So the structure of these, they are nonpolar. And so again, you're not seeing a charge on these 2 legs or tails. And since they're nonpolar, they dislike water. And so you're going to find them, remember your cell membrane is a phospholipid bilayer. And so if you're looking at this picture at the bottom, these nonpolar tails want to get away from, if this side was the exterior of the cell and this side was the interior, those hydrophobic tails want to get away from the water and attract to one another. And so these structures, since they are also hydrophobic, they're really going to be important in regulating what does get to pass through the cell membrane. And then the other thing that I want to point out, one of these tails is saturated. So if you look at every carbon, it's saturated with a hydrogen. So it's a straight line. And then the other one has a double bond here. So it's unsaturated. It has a little bit of a kink. And so when you think about the cell membrane, you do want those phospholipids to create a barrier, but you don't want them to pack too tightly together. And so by having that one leg kinked out, then it still allows what needs to get into the cell to come in very easily and what needs to leave to exit very easily. The other portion of a phospholipid, what makes it unique in its structure, is that it does have this polar head, and that's due to having a phosphate group, which has a negative charge. You also have a positive charge up here. And so since it is charged, we're going to call it polar. And if you remember, polar things like one another and water is polar. So these are going to orient themselves facing the water exterior of the cell and the water interior of the cell. All right, really fast. I just want to show you a different lipid molecule, and that's cholesterol. And the only reason is that you're going to find cholesterol embedded in between those phospholipid tails of the cell membrane, and that's also going to help maintain the fluidity of the cell membrane. So I'm hoping to wrap up with the cell membrane in a little bit. So again, I just wanted you to see another molecule in this lipid category, different structure and different function. All right, so moving on to proteins. Proteins, I feel like you would need to specifically, you should recognize that that small building block of a protein is amino acid. And so I would want my students to use that term. There's 20 that are found in nature. And those monomers are held together by a specific covalent bond. So if, on test day you're nervous and you say that amino acids are held together by covalent bonds, you are correct. But sometimes we can be more specific. So going back to chat, can you guys tell me the specific bond that holds two amino acids together? I don't know if I can see that anymore. Are you getting answers, Lou? There we go.
And just to jump in,
Yeah.
None of the bonds actually in the structure shown on your PowerPoint are this bond. You're referring to the bonds between two different amino acids.
Right.
Correct?
Yeah, just for right now, yes. Looks like we're getting it. Anybody else want to jump in? All right, so peptide bonds are the specific. And so if we link many amino acids together, then that polymer, we would call a polypeptide chain. And so we're not going to get into these details today, but I do just kind of want to tie in that particular sequence of amino acids is determined by a particular gene that you inherited. So what I have at the bottom is a picture of just a generic amino acid, again, showing you the elements that they're composed of. And so any amino acid, if you're seeing a picture and you're trying to figure out what they're showing you, look for this if it's an amino acid and therefore a protein. And so it starts with a central carbon. And so that carbon is going to have an amino functional group on one end and then a carboxyl functional group on the other end. So you may see this, instead of the negative sign, you would have a hydrogen there. And then off from that central carbon, you have a hydrogen and then an R group. And so the R group is what makes the 20 amino acids different from one another. And so here's a picture. You definitely do not have to memorize memorize these or know any details, but I think on a question, they may give you some of those details and some of the vocab that we've already discussed, and then you need to think through the situation. So at the top, all of these amino acids that have the green highlighted R group, those are nonpolar. And so remember, nonpolar means hydrophobic, and that means they do not like water. So how is that going to affect the structure of the molecule? These guys are going to want to find themselves in the middle. So however the polypeptide chain orients itself in the end, these particular amino acids want to find their way in the middle so they're not exposed to the watery environment of the cell. Same thing with the polar side chains. These guys do like water, and so they're going to be on the exterior of that molecule, whatever shape it may be. And acidic and basic, they also have charges, so they also would interact with water. So also to kind of just tie in what we discussed earlier with the generic idea of attaching our monomers, here's how we would attach two amino acids together. And so at the top, there's your two separate amino acids. They're showing you how water would be removed, and then the bond between the carbon and the nitrogen of two different amino acids. There's your peptide bond. All right, so proteins that actually do work in your cells, there's several levels to get to that final structure that allows them to do their particular job. And so to go through, the first level of structure in a protein is the primary structure. And so this is that particularly unique sequence of amino acids. And if I was to describe it, those amino acids are held together by peptide bonds. So again, to make you guys think and see if you can tie in what you learned earlier, what organelle does the sequence of amino acids get assembled? So going back to your cell biology, at what organelle will we create proteins? All right, we got a couple answers. Anybody else want to participate? All right, good job. I think you're all in agreement on ribosomes. I've kind of already mentioned this and gave that part away. And so the overall message that determines that sequence comes from your DNA. All right, oops. So going back, so after the particular sequence of amino acids is created, now different portions, different amino acids can interact. And so there's two possible shapes in the secondary level of structure. And those can either be an alpha helix or a beta pleated sheet. So an alpha helix is, think of your DNA except one strand. So it is kind of that curly Q shape. And then beta pleated sheet is kind of like a zigzag. And so how does this shape possible? This is through hydrogen bonds occurring between a hydrogen of an amino group and an oxygen of a carboxyl in nearby amino acids. And I do have a picture for you so you can see this. So now that we have this secondary structure, our R groups can finally interact. So all those different groups that had different properties and made each amino acid unique, those can interact with one another. And so that's going to allow this level of structure to occur. If you were describing what's going to kind of maintain that structure, all kinds of interactions that we've discussed this year. So hydrophobic interactions, van der Waals, hydrogen bonds, disulfide bridges, and so forth. And then for most proteins, they usually consist of two or more of these polypeptide chains. And so those, again, any of the interactions mentioned in the tertiary group can occur here to allow two polypeptide chains to interact. And so this is what it looks visually. Starting at the top, you have your amino acids linked together. So one amino acid beside another. So peptide bonds are keeping that together. And then due to hydrogen bonding, we either get that beta pleated sheet, so kind of zigzag, or the alpha helix. Those are hydrogen bonds keeping that shape. Now, because of these shapes, our groups have a chance to interact. And so if you think back to some of those examples, I said hydrophobic ones would want to orient themselves to the middle of the tertiary structure. And so if you could imagine somewhere in the middle there, all those hydrophobic amino acids would find their way there. All the hydrophilic or charged would find themselves kind of in the exterior of this polypeptide chain. And so then to finally get to a functioning protein that's going to do some work in your cell, this polypeptide chain would interact with a totally separate polypeptide chain. And then it can actually do some work for you. So I wanted to tie this one in so that we could get some writing practice. So your test this year is 2 questions that you're going to have to write and explain. And so again, just back to the levels, you have your primary structure that leads to your secondary, alpha helix, tertiary, and then for hemoglobin, this protein is the protein in your red blood cells that helps carry oxygen throughout our bodies. And so it takes 4 polypeptide chains to do that efficiently. And what you're going to be asked to do is, what happens when something happens to this shape? So what happens to its function then? So this is a great example, ties a lot of our units together that we have discussed so far. And so what could cause a shape change is a mutation. And so you guys possibly learned about sickle cell anemia. And so back to our DNA, remember I said DNA sequence determines what amino acids get linked together. And so here in the 6th position, glutamic acid would make a normal hemoglobin. And so this is what your red blood cells look like. And then if there is a point mutation, so just one single letter got changed in DNA, at that 6th position, valine would have been brought in. And so you get this sickle shape. It's kind of a stretched out, flattened shape. So obviously it's not going to be able to carry oxygen as efficiently. And then it has a greater risk of clotting together and creating clots in your blood vessels. So I try to create a free response short answer to mimic what you guys are going to see on the 2nd question of your exam. And so they've already given us the format. So on your test on May 18th, question number 2 is going to ask you to describe something, explain something, make a prediction, and then justify your prediction. So I try to create one. I am not great at creating questions and I'm not great at writing, but hopefully I'm giving you a pretty good idea of what to expect on test day. So here's what I would like you to do. I think this is too much for us to kind of do in the chat room, but if you have pen and paper handy, then on test day, how I would think through this, I think when you read a question, automatically stuff pops in your head that you think is what you should mention. And so that's how I would start. I would read, starting with part A, jot down some things that I feel like I need to put in my final response, and then do that for all parts. And so the reason I say that is because sometimes I think we look at A and we're like, oh my gosh, I have no idea what they want from me or how to answer this. But then later on, like in C or D, they give you a little bit more clues of how to respond. So I'm going to read through. What I would like you to do is kind of just jot down in your head so that you're actively thinking about this. What would you say? What do you think would be important to say? Obviously, I don't have 15 minutes to wait on you guys to write a good free response, but take the time, jot down what you think would be a good answer, just, not complete sentences. And then I'll go over what I think would be a good answer for this question. So for part A, you're asked to describe the primary structure of a polypeptide chain. And I'm asking you to be sure to identify the bond that occurs between 2 amino acids and how that bond can actually form. For B, I want you to explain the difference in how a hydrophilic R group would position itself in a particular region of a polypeptide chain versus a hydrophobic R group in determining the tertiary structure of a polypeptide chain. And then for C, I want you to make a prediction. So in normal hemoglobin, the 6th amino acid, so if you think back to that previous slide, is glutamic acid. And so glutamic acid has a negative charge and is hydrophilic. If a point mutation occurred at that site, valine is brought in at that 6th position instead of glutamic acid. Valine is hydrophobic. And I want you guys to predict the effect of this change on the structure and function of hemoglobin. So I want to stress something. These task verbs, the describe, explain, predict, those are typically in bold and they get your attention. On the other hand, sometimes they're going to squeeze in an and it'll be in all caps, but it's not in bold. But to get this 1 point, you have to talk about structure and function. So if this change occurs, what happens to the structure and function of hemoglobin? And then for part D, you just justify your prediction. Why do you think this change is occurring to structure and function, if you think there is one? All right, so I'll give you a little bit of time to continue jotting down your thoughts and then we'll go over it. Lou, really fast, I forgot to look at the clock. I have no idea time-wise where I'm
We're about
at.
halfway through, Mary Beth. You're doing great.
Okay. And while you guys finish up, something that you can do, after this is over, you could go back and then, I'm asking you just to kind of jot down what you would want to include, actually practice writing it out. And I think that's where we get really good at free responses is, putting it into words. Does my sentences make sense? Have I completely answered the question? Even though we've talked about it, once we're finished with this, could you put it in your own words to make sense? All right, so I'm going to go ahead and move on. So for A, I did go a little bit further, and so I tried to underline what I felt like truly would have earned the point here. And so the primary structure consists of a sequence of amino acids held together by peptide bonds. And so I asked you to describe the structure and then tell me how those bonds actually occur. And so these bonds were formed through dehydration synthesis. And this is where I went a bit extra. And so because I knew this, if you didn't know this, don't worry about it. So how does a dehydration reaction occur? It's a hydroxyl group's removed from the carboxyl end of an amino acid, and hydrogen is removed from the amino end of an amino acid. So the removal of water allows that peptide bond to form between the growing chain of amino acids. For part B, I wanted you to explain to me how a hydrophilic R group would orient itself in a polypeptide chain versus hydrophobic. So hydrophilic R group interacts well with water, and so it would position itself on the exterior of the molecule because the environment of the cell is mostly water. And the hydrophobic R group hates water and will orient itself to the middle of the molecule to avoid having to interact with the water environment of the cell. For part C, you were asked to make a prediction. So glutamic acid was replaced with valine. And so glutamic acid was hydrophilic, valine's hydrophobic. And so valine is going to cause the protein to fold in at that particular portion of the polypeptide. And so the structure has now been changed. And because of that, the function will also change. So in my prediction, I've told them what I think is going to happen to the structure as well as the function. And then my justification, I just kind of go into details of why I think that's going to happen. And so for D, as stated above, glutamic acid's R group allows it to interact with water and would be found on the exterior portion of the polypeptide chain. If a mutation replaced glutamic acid with valine, valine wants to hide from water, so it goes inward away from the water. This change in shape or structure of the protein no longer allows hemoglobin to carry oxygen efficiently. So I'm still stressing, I'm telling them the change in shape and structure. So I'm telling them it has changed because of this mutation. and it no longer allows hemoglobin to carry oxygen efficiently, and that is the function of hemoglobin. But you can go further. I wanted to just stress that I knew structure and function was connected, and so cells in the organism will not get the oxygen they need to survive, and the change in the structure of this protein does not allow it to function anymore. So I just kind of stressed that I knew change in structure causes a change to the function.
And Mary Beth, may I interrupt for a second here?
Yeah, absolutely.
And this is a non-biologist speaking, but my understanding is this would be a mutation. The students would not necessarily need to state that this mutation prevents oxygen from being carried. And in fact, I would suggest that mutations drive evolution, and that sometimes you'll get a mutation, a change that would be better and beneficial to the organism. And then you can talk about natural selection and tie all of this wonderful biology together.
Right.
But just to say that the structure would change, and therefore it would likely to have an effect on the function.
Yes.
Mary Beth, can you go back? We have a question in the chat room. Can you go back to the part A for just a moment?
Yes. Okay.
So let's say a student read the question and understood, they remembered a term associated with this that they think is associated with this question. The question that the student asked was, would you get credit by mentioning the term in the definition but not explaining it? So maybe if you could talk just a few minutes about, how students need to construct their responses based on what they know as they're reading the free response questions.
So like defining what dehydration was, is that?
Yeah, so as the student looks at the question for part A, and I don't remember what the question was specifically. I'm assuming it's I see the answer, but how do they, so when it says describe the primary structure of the polypeptide chain, I think the student's trying to say that they understand some of the definitions and words associated with that, but suffice to say that they shouldn't just put down a term and a definition, but can you give them some guidance on how to construct their answer?
I think sometimes they could possibly get lucky. So like if they weren't completely sure what the structure was, or they didn't know fully, but they do recognize a term, then I think absolutely define a term. So, I think they, it just depends on the question. Sometimes they could earn points by explain that they know what that term means. In this case, they were asked to describe the primary structure of a polypeptide chain. So they would need to say that it consists of amino acids linked together through peptide bonds. And I gave them a little bit more guidance here. And again, I made this question up, so I'm not sure that it's the best, question to refer to as far as AP level, but I kind of just wanted to give an example based on what we're discussing here. But I have always told my students, if they have no idea what the question is truly about or where they should go, then it absolutely doesn't hurt to define a word that they recognize in that question.
Okay.
Does that help?
We'll watch the chat and see.
Okay.
But I think so.
Okay. Anything else here?
Well, I think historically the AP Biology pre-response question has really insisted on having sentences and paragraphs and not bullet points.
Right.
That may be relaxed with the new format of the test this year, but I think diagrams are always welcome. So if one were to sketch out a polypeptide and just amino acids and even a peptide bond, if you remember your chemistry that well, that would certainly help. But I think the sentences and the descriptions are really what's called for.
Yeah. All right, so I did want to just go back and based on the answer, kind of show you again, one more picture specifically to sickle cell anemia. And so here was our normal hemoglobin. And then here you're seeing where that 6th amino acid was changed. And then based on that primary structure, we get our secondary and tertiary structures. So they're showing you, remember glutamic acid was polar. And so you can kind of see it is full, all parts of that polypeptide is kind of exposed to the water environment of the cell. And then valine, if that was the mistake, then that part is trying to get away from water, and you can see it's folding in there now. And so at the top, you have your normal hemoglobin compared to sickle cell, and you can see the function of that molecule is to carry oxygen and to stay separated from one another. And then with the mistake, those tend to aggregate together. They don't carry oxygen as efficiently, and they can, risk clotting in your blood vessels. So there's just a visual to our answer. All right, besides mutations, I did want to mention some environmental conditions that can affect protein shape. So again, on these AP questions, they're probably going to mention some protein that you've never heard of, I've never heard of, but if they're telling you it's a protein, all this stuff that we've gone over still applies. And if it's a mutation or any of these environmental conditions are being given to you, then you should be thinking, how does that affect the protein shape and therefore its function? So a change in temperature, specifically an increase in temperature, is going to or can alter the shape of a protein. Change in pH, either direction, so it becomes more acidic or more basic, and then an increase in salinity. So the salinity one is usually, if you think about those R groups again, if they had charges and the R groups were interacting with other R groups of that polypeptide chain, if the environment now has more charges surrounding that molecule, now those R groups have more things to interact with, and that would cause your polypeptide chain to unravel, so it loses its shape and therefore loses its function. And so we would describe a protein as being denatured if it's lost its shape and its ability to function. So here's some examples for those of you that have curly hair. That's usually a result of disulfide bridges holding that curl shape to your hair. Your hair is a protein. And so when you flat iron it in the morning, you want straight hair, you are temporarily disrupting those bonds. So you temporarily disrupt its shape, and it, not that it particularly has a function, I don't guess, maybe keeps you warm a little bit. It probably wouldn't affect that particular function. Cooking an egg, you all can see that for sure. So the liquid form when it's not cooked and then you cook it, you have definitely changed its shape. High temperatures when you're sick. So a little bit of a temperature, 99 to 100 is good if you're sick, because that is going to affect the proteins of the bacteria or the virus that's causing your infection. And it doesn't totally hurt your proteins that are still helping your cells function. And so then we can go further. This is what happens in the summer when children or pets are left in a locked car. How does that hurt them? The temperatures, I forget what they get up to, but it's so hot, then that affects the proteins in those organisms, and so they no longer can do their job. All right, so again, I kind of just wanted to stress that proteins, the function of any protein always goes back to that very primary structure and how those monomers, those amino acids, were assembled. And so here's some different proteins that do different jobs in our cells that we've discussed this year. I want to spend a little bit more time, if we have it, on enzymes and then on transport. Still good? Okay. All right. So I wanted to give you a picture. This is not particularly catalase, but I wanted to give you an enzyme, catalase. I use this in lab as a demonstration for my students to understand how enzymes work. And so I'm going to give you the information that catalase, its job is to break down harmful hydrogen peroxide into water and oxygen. So I've kind of given you everything that you need to know could you label A through E with these components? So could you identify the parts of this chemical reaction, giving you that catalase is the enzyme, and its job is to take hydrogen peroxide and break it down into water and oxygen. So let's try this with chat. I think it'll work okay. If you'll just A, what do you think A is out of those components? B, C, D, and E. All right, so we've got Trevor's participating. There can be a special term for C. We'll talk about it. I don't know that it necessarily is important to know in this particular example, but okay. So we'll go ahead and talk about the parts. So if you look at this picture, the part that's not changing, that's your enzyme. So if they're mentioning some enzyme that you've never heard of, a good clue, if they're not telling you it is an enzyme, if the word ends in ASE, that's a big clue that you're looking at an enzyme. And so the great thing about enzymes, most enzymes that I think will be discussed in questions are proteins. So again, you can think about all the things that we've discussed and what affects their shape, apply that to an enzyme. But the great thing about enzymes in them facilitating the chemical reaction is they don't get used up, and their shape doesn't get altered. So if you look all the way through, this B and D, that's our enzyme. So it stayed the same, it facilitated a reaction, and we get to reuse it over and over again. A is our substrate, and so they have the perfect shapes for one another. So on B, this would be the active site, so that's where our substrate fits and interacts. C, a lot of you did get this, that's our enzyme substrate complex. And here's what I kind of always think of when I see this picture. The enzyme's job is to kind of create this little environment that's going to either break bonds in the substrate or create bonds. So in this case, we are wanting to break A down. And so the enzyme at C is going to kind of hug around the substrate. So when they come together, the enzyme will slightly change shape to hug around. And what this makes me think of is when I was little and I'd see my uncles, they'd grab me up and it'd be like a big bear hug, and I always felt like they were going to break my backbones. That's what's happening here. It's kind of creating enough of that force to break the bonds in that substrate molecule. And so we end up with E, which is our product. We wanted to break hydrogen peroxide, A, into water and oxygen. And so there's our two products. Number two, again, we can go to chat. So with this, going back to our terms we talked about earlier, would this be a catabolic or anabolic reaction? Anybody else want to participate? So remember, I said C for catabolic, C for cannonball. So cannonball is kind of like breaking. So this would be a catabolic reaction. We want to take this larger A, our substrate or reactant, and break it down into two smaller components. So that would be catabolic. Do you guys remember how enzymes actually speed up chemical reactions? So I haven't covered that today. I just kind of wanted to see if you remembered it from your studies this year. So again, go to chat. See if you guys remember how enzymes speed up chemical reactions. All right, so we've got some good answers. So activation energy is lowered, so good job on that. So somebody's asking, Angela, hey, sorry I missed you this year. What exactly is activation energy? So that's the energy required to convert the substrates into the reactants. And so a lot of times you may see graphs showing you this. And so sometimes they'll kind of look like a big hill. So if you drew a big hill and that's the typical energy, that's your barrier that you've got to overcome from going from a substrate to a reactant. So you have this huge hill that you've got to overcome. Enzymes, think about having the ability to pull that hill down. So if they pull that hill down, it's the same reaction, but obviously I can run a lower hill faster than I can run that big, tall hill. So that's kind of how I think about it. Oh, I'm sorry, I'm just seeing you now. So anyway, last question, just to tie in our structure and function. If the active site shape changed, so active site is here, what would happen to catalasis function? So again, let's go back to chat, try this one out real quick.
And while the students are doing this, Mary Beth, I'll say just a couple of things. We're just about at our time. It's 11:53 on my watch. That's terrific. Let me also put a plug in that these enzymes and substrates are attracted by what a chemist would call intermolecular forces. And that's those subtle pluses and minus charges on the surface of the enzyme. This is why enzyme or the shape of the proteins, these enzymes are so critical and so remarkably specific that enzymes that put DNA together or break down carbohydrates are incredibly well tuned, and just changing one amino acid, as you can see from your example with hemoglobin, could have huge consequences for the organism. But those of you who are in AP Bio students, maybe we'll see you in AP Chemistry. Intermolecular forces is one of the central topics there. And but thank you. So we're just about 5 minutes ago, or whenever.
Oops, I kind of only hit the last thing. So some of you just told me about the substrate. So the question was asking you what would happen to the function of catalase. Some of you got it. And so you would want to mention that it could no longer function correctly. So if the active site changed, the shape has changed, what's going to happen to its function? It's not going to be able to do its job, which is to convert hydrogen peroxide into water and oxygen. So good job there. Do you want me to go ahead and just wrap up, Lou?
Sure, we've got a few minutes.
This last slide that I gave, and you guys could work on after this, and if you're looking at a later time, this is something that you might see on question 1. And so I've given you some information about 2 different enzymes, temperatures being changed, and they're looking at the rate of the reaction. So what does this temperature change do to the rate of the reaction for these enzymes? Same thing, what happens to as pHs change, what happens to the rate of that reaction to these two enzymes? So let me go ahead and go to, I think 1 to 3 points could be on analyzing some data. So could you describe trends or patterns? Could you describe relationships between those variables? And I wanted you, if we had the time, to focus on just the black lines for temperature and pH. So I am going to go ahead and show the answers. So if you want to go back and look at this later, you can do that and see how I answered this. So there's answers to those two questions for the temperature, and then there are answers you could pause and kind of look at later for the change in pH. All right, guys, thanks again for joining us. I hope you've learned something about structure and function. Best of luck to you on the AP test. Stay positive and stay healthy. Thank you.
Thank you very much, Mary Beth. That was terrific. I learned a lot. I'm ready for the exam.